My name is Anwar Hossain. I'm a PhD student at the University of Melbourne. Today, I'm going to present estimating and conserving patterns of grasshopper diversity and richness in Western Australia. Insect conservation is often limited by a lack of data. Most of the reports of insects decline has come from North America and Europe, with only one report was found from Australia, which is based on managed honeybees. Evidence suggests that insects are also declining in Australia based on historical times in the recent past at smaller spatial scales. However, the status at continental scale is unknown due to a lack of baseline data at large spatial scales. For grasshoppers in Australia, extensive information is contained within field notebooks from surveys conducted between 1936 and 1989. So here is an example of a field notebook and how they have presented the information from their field collection. Here is the highlight of the first collection site. We can see that the survey was conducted in the September of 1968, and they were heading from Mildura to Port Augusta. The collection points were indicated by car odometer. At each collection point, they systematically recorded the soil and terrain type, vegetation condition with common plant species, collected grasshoppers, and their life stages. The collected grasshopper specimens were preserved in the Australian National Insect Collection, and they were associated with the car odometer of each location. In summary, field notebooks give us the following information: date of survey, location and route of travel, terrain type, soil type, vegetation condition, collected grasshopper species, and their life stage. As we heard in the plenary today, museum specimens have valuable data associated with them. We took this opportunity to study the grasshoppers in Australia using field notebooks. Specifically, we aim to use those notebooks to develop species richness and turnover maps for grasshoppers in Western Australia. We aim to identify center of high endemism from field observation and apply results to assess the representativeness of protected area network in Western Australia and discuss their conservation planning in the Western Australian context. For data preparation, we transcribed field notebooks for surveys conducted in Western Australia. Then we collected grasshoppers' historic location from those notebooks using historic topographic maps and Google Earth. This was then validated by another person. To avoid a biased model due to the inclusion of sites having very low species count, we aggregated the whole area of Western Australia into grids of 50 kilometers. We collected grid centers as the location for surveys that fall within that grid. In total, we collected 32 predictors covering bioclimatic, soil, NDVI, digital elevation, and topographic wetness index. For bioclimatic variables, we extracted values as a long-term mean of historic temperature and precipitation. To analyze patterns of species turnover, we fitted a generalized dissimilarity model to analyze this. Generalized dissimilarity modeling is a statistical technique for analyzing and predicting spatial patterns of grasshopper in community composition across large regions. In this study, important predictors were selected using a backward elimination process in which the final subset was selected having a p-value of less than 0.05 for all predictors in the model. Finally, a subset of 7 predictors were selected to be included in the model. The final model was fitted using the selected predictors and using the GDM package in R. To analyze species richness in Western Australia, first we fitted a regression model using all predictors. We then eliminated the worst predictor based on lowest t-value. The process was repeated until a subset of predictors left in the model having a p-value of less than 0.05. Finally, we predicted species richness using the selected subset of predictors in Western Australia. To identify centers of endemism, we used weighted endemism approach, in which higher scores are assigned to species with smaller ranges. For example, if the distribution of a species is confined to one cell, it was scored 1 for that species in that cell. For a 2-cell distribution, the score was 0.5. For a 10-cell distribution, the score was 0.1, and so on. The final endemism score of a cell is calculated as the total score contributed by all species in that cell. We checked the correlation of weighted endemism with species richness, and if they are highly correlated, we measured the corrected weighted endemism, in which is calculated as dividing weighted endemism score in each cell by the richness score of that cell. This is the predicted compositional turnover of grasshopper species in Western Australia. Colors represent gradient in species composition derived from multi-dimensional transformed environmental predictors, in which similar colors are expected to contain similar grasshopper communities. We identified that at least 4 distinct biogeographic regions of grasshoppers are present in Western Australia. These are Kimberley, Pilbara, Southwest, and the arid interior. Based on our regression model, we found that species richness was highest in Pilbara and Kimberley and lowest in southwest of Western Australia. The figure on the right shows the coefficient of variation based on 100-fold cross validation. Red dots on the map indicate the location of surveys. The coefficient of variation of models show that high model uncertainty of up to 2% was observed in areas with no field surveys. In this figure, white cells are unsurveyed, gray shades in the background represent protected areas, blue shades indicate indigenous protected areas, and pink indicate private nature reserves. Blue lines represent the boundary of bioregions. This figure shows the endemism of grasshopper species with a distribution range of 1 to 4 grid cells. Red points on the map indicate locations of 1 to 4 cell endemics that occur within protected areas. We found that 48.6% of species had 1 to 4 cell distribution in Western Australia. Only 26% of them are located in protected areas. The figure on the right shows the corrected weighted endemism in Western Australia. This highlights that endemic grasshoppers are distributed throughout Western Australia. Here are 3 take-home messages. We have identified areas that require conservation attention. For example, we found that Pilbara region has the highest grasshopper species richness, as well as a distinct compositional pattern to its neighboring areas. Also, the area remains underrepresented in the protected area network. Large number of grasshopper species are very rarely encountered, with only 26% species that are 1 to 4 cell endemics falling within the protected area network, although it is uncertain whether protected area provide any protection to endemic grasshoppers. Field notebooks serve as important source of baseline data to augment our understanding of a lesser-known taxa such as insects. Additionally, field notebooks provide us an opportunity to assess how a really important part of our invertebrate fauna has changed in distribution and abundance over time. I'm very much grateful to my fantastic supervisors, Michael Kearney and Hossain Md, and my collaborators, committee members, and volunteers. Also, I acknowledge the traditional owner of the land on which I work and pay my respect to the elders past and present. This study was funded by the Melbourne Research Scholarship and the Research Training Programs. Please let me know if you had any question. Thank you very much.
Thank you. It's a great analysis of historic field notes, and just a comment from me. I'd I'd hate to think how many historic field notes are discarded at the end of someone's career that you have no idea, or at the time, how useful they will be in future studies, particularly in climate change and habitat loss. So well done on that interesting analysis of Ken Key's grasshopper projects. Yeah. Do we have any? Yeah. Do we have any questions? A comment from Sonu. Yeah, great talk, Anwar. Any plans to extend this analysis to other states?
Yes, so basically the main drawback here is it takes a huge amount of time to process, to transcribe and find the geolocation. Huge amount of time it requires. So for that was for my PhD that cannot suffice. So we thought, okay, which state we can select. So in future, after I have finished PhD, and yeah, definitely we can do that to extend to other states.
Okay, thank you. I see Martin Steinbauer has his hand up for a question or a comment.
Thanks for that talk, Anwar, very interesting. Just curious, I might have missed it. A lot of the grasshoppers are very good dispersers. So how does the model account for that?
Okay, so we did not model how the dispersal ability of grasshoppers. So these models are based on the occurrence points where they have been detected. So we just took that historic occurrence locations as input data in our model. So for good dispersal, for example, some pest species, locusts, and then that will be different kind of analysis. So we just here interested to check what the richness areas of grasshoppers be, and how well, how many occurrences they have, and okay, and then get their turnover so that we can get an idea what's going on there. And then if we want to extend this analysis for conservation perspectives and revisit the area, so this will help.
Yeah, I wasn't just thinking of the locusts. A lot of the regularizing species, I was thinking of quite a few of them are quite good flyers and turn up at all sorts of places after rain, especially in the Pilbara, etc. So yeah, I look forward to seeing how you deal with that. Thanks.
Thank you.
There's just another quick question that is related to Martin's, and that's come from Steve Cooper. Have you any plans to revisit any of Ken Key's historic sites to see whether the species are still present?
So it is possible to visit some Ken Key sites. In 2019, we did a pilot study from areas from Canberra to Brisbane, and we selected sites that were conducted in the same time period of the year, and we found lots of thing happening in terms of grasshopper distribution. For example, for one species, Acrida conica, in that time they found only two locations, but we found that species dispersed in a lot of locations. Also, they didn't find in some areas no species. We found many, and also we found that lots of changes in vegetation. So yeah, definitely it's good looking into in future to see what's happening here. Maybe it is really important to do some resurveys into the areas so that we can get a good understanding of what is happening there. Thank you.
Okay, thank you, Anwar. We're right on time now, so we might move to the next